right, so what I'm going to do is I'd like to I hope you're kind of interested in this because some of you who you go to school and you say, I'm going to be a pre-med or pre-vet or biology. They're going to make you take biology and chemistry probably at the same time. So if you have anything that has to do with the medical field or anything like that, they'll make you take biology and chemistry. And so let's take a look at these. What are natural polymers? We know that the ones that are human-made are things like nylon and Dacron, and we make plastics all over the place. Those polymers, rubber, we know how to make polymers. But isn't it interesting that God already gave us the way to do it? If we just known a little bit more about biological systems, we'd have known that was already there. I always think it's interesting to think DNA. What makes your DNA different than an ant's DNA? Why is your DNA? What's different about the ant's DNA? I had a little boy. He's in like 6th grade. He came in on Friday, and his mom works here. But he just happened to come here, and he said, our teacher was talking about atoms, and what I don't understand is how can the atoms be different when all the protons are kind of this, they're made of something smaller than that anyway. And it wasn't the fact that the protons of carbon aren't any different than the protons of magnesium, are they? So what makes magnesium different than sodium? It's not the protons. What is it? It's the combination, isn't it? And the combination of 11 protons allows you to have 11 electrons, and 11 electrons are in certain locations, aren't they? And so the chemistry of 11 electron atom is going to be different than one that has 12 electron atoms. See that? It was the combination that did it. And we talk about flame tests. He's only like 6th grader. And talk about flame tests when you excite the atoms and they jump from one energy level to another. And this one will give off this color. This will give off another color. Anyway, he went home this week, and I guess his mom made me a little note, wrote me a note, and said he was really, really excited. What about this thing here about DNA? What's different about your DNA than and we'll get to DNA here. What's different about your DNA and ants' DNA? Is your DNA made of deoxyribose sugar? It is. Is it made of phosphate groups? It is. Those are the same. Do you have bases, nitrogenous bases that are A, T, C, and G? You do. So does the ant. So why aren't you an ant? And again, it's not the fact that your adenine, guanine, cytosines are different. It's the sequence that makes them. See that? And so when we see this, same thing here, we're going to do polymers. What's the difference between having a sugar and 2 sugars hooked together, 3 sugars hooked together, 1,000, 10,000 sugars hooked together? They're going to act differently, aren't they? And that's really what chemistry is about. It's all these combinations. What makes the combinations act differently? So, you didn't die. That was important. So, are you okay on those? Is it coming back to you? All right, here, I'll take it. All right. Okay. Well, anyway, we had on our handout sheet, what I'm doing is starting at page 1025, natural polymers. And the one I want to start with is probably the most important one if you're a biology person. The word from Latin comes from protein. Now, I don't know Latin, but help me out. Have you ever had the word protein in Latin? How about proto, like prototype? What is proto? Do you guys have proto? Okay. Anybody know what proto means? How about prototype? The first, isn't it? A prototype plane, that may not be what the plane's going to look like when they actually start manufacturing. You see that? But it's a prototype, isn't it? Well, protein means of first importance. And so if you ask a biologist, what's the most important organic molecule, because water's not organic, but the most important one, they'd probably say proteins. So let's take you back. Let's go back to 9th grade. No, I don't want to go there. Okay, let's take it back a little bit, ninth grade. The four most frequently found elements found in living things are Chan, aren't they? But what are the four most important organic molecules found in living things? And they are proteins, carbohydrates, lipids, and nucleic acids. Do you remember this at all from ninth grade? Now, of these four, three of them are polymers. Three of them are polymers. You get that? And lipids are not. Lipids are fats, and they're made of fatty acids and glycerol. I probably won't talk about them hardly at all. But these are actually polymers. So there's going to be a monomer that keeps repeating itself like that. All right. So, excuse me. So anyway, let's get my handout sheet out. So proteins are the most important of the, biologists would say they're the most important. Now, there are two kinds of proteins, and I'm not your biology teacher, but I'll show you anyway. Proteins are great at building things. They make up structures. They can make up cell membranes. They can make up ribosomes. They can make up lots of things. The proteins are great at building. You can build with proteins. Your muscles are protein, right? Your hair is almost all protein. So your fingernails. Anyway, protein can be used as a building structure. But there's another type of protein which is not, excuse me, which is has a unique job, and those are called enzymes. Now, do you remember that from ninth grade? So you got structural proteins, and then you have enzyme proteins. Now, enzymes are catalysts, aren't they? They cause chemical reactions to take place, but they themselves can go and do it again. Let's go do it again. They themselves are not altered, are they? That's called a catalyst. So if you were to read about proteins, it'll say structural proteins, and then they might have enzymatic or enzyme proteins. Okay. But what do they have in common? Well, polymers are macromolecules made up of long chains of monomers, and sometimes they cross-link with other strands. Synthetic polymers can be plastics, rubber, styrofoam, PVC. We talked about that before, nylon, Dacron. Monomers become polymers through either addition polymerization or condensation. Now, I know it's Monday morning, and you still can't believe that Meghan Markle said all this stuff. I didn't watch it, by the way. I did not watch it. But what is the difference between addition polymerization and condensation polymerization? I know that one of you had groups in here do that. Which one? Do you know?
For addition, you don't lose.
Say it.
Double bonds. Is that the one?
Yeah, the double bonds become single, but you don't lose any what?
So they connect to the
right?
close by electron.
And you don't lose any
Hydrogen.
atoms, period. You don't lose any kind of atoms, do you? But what about the one that was called a condensation polymerization? I forget who did that. Maybe, was that Jeb? What do you lose in a condensation polymerization? Such as water or HCl or something like that? That means maybe I take an H off of one of you and an OH off another to form water. All right, so there's your general idea of what a polymer is. And here's addition polymerization. Here's condensation right here. And so here's a monomer. Here's a monomer. What do you call it when you put two monomers together? A dimer. What about three polymers together? Trimer. And what about 1,000 of them? What? Polymer. Everybody get it? Oh, I think I get this now. All right. So let's start with proteins. Now, proteins are long chains of monomers called amino acids. Are you with me on that? You remember that at all? Okay. Now, what's amazing to me is amazing. If I analyze that blade of grass and that dandelion, and that squid and that mountain lion, if I analyze their proteins and that fly, I would find out that all their proteins are made of amino acids joined together as a polymer. Get that? So how many different kinds of amino acids must there be? I mean, I got the wing muscle of a fly. That's different than the cow's muscle. That's different than the hoofs of an elk. What's different about proteins? There are millions and millions of different kinds of proteins. But how many amino acids have we ever found? Only 20. It's almost like there's only 26 letters of the alphabet, but how many? Have you ever seen Mr. Powell's dictionary he bought at the, did he show you about that? I saw, it's an unabridged dictionary. There's only 26 letters in the alphabet, but how many words can you make? More than that. That's when they printed it. So there are only 20 amino acids. And so how many different ways can you rearrange them? And by the way, I didn't say you had to use all 20. Could I use the number 1 5 times in a row? I could. How many ways can you arrange amino acids? How long? I didn't tell you how long. All right, a lot of amino acids are, I would say, 60 to 70 would be the smallest chain of amino acids, but it can be much, much bigger than hundreds of amino acids long. So maybe a small protein might be 60 amino acids long, and a big one might be 560. So that makes them different, doesn't it? Okay, so there are only 20 kinds of amino acids. Now, when you have proteins, you'll hear this, sometimes you'll hear stuff like this. Hemoglobin, myoglobin. Have you heard that? So if you hear something like that, that's probably a some kind of a structural protein, and they like to use that at the end of a word. But what if the protein was an enzyme? What do they always put at the end of an enzyme? You say, well, I don't know. I don't remember that. I don't remember that in 9th grade, and I haven't read this yet. And they would use ACE. So if someone said, by the way, do you know that OSE at the end means sugar? So if I say sucrose, glucose, do you know that means sugar, don't you? But what would sucrase be? Sucrose is a sugar. What would sucrase be? An enzyme that knows how to break down sucrose. In your spit, you have this stuff called amylase. And I know there's an enzyme that breaks down carbohydrates. What about protease? What about protease? What enzyme would that break down? What do you think it might? Break down what? Proteins, right? Okay. So how about DNA polymerase? And I said, well, you're sounding pretty scientific today. So DNA polymerase, you'll learn later that DNA is a polymer. It has to be put together, doesn't it? Well, who does that? DNA polymerase. RNA polymerase. So either the enzyme will end in ACE, or it might be a structural protein that might end in something like N. Okay, now one thing that is you'll find out as you read this is that I have this long chain of amino acids, and that's called the primary structure. We're going to get to that, but there's a primary structure, meaning what's the sequence? So what makes your leg muscle proteins different than a dog's leg muscle protein? And it's the number of amino acids and the sequence. It's the sequence of amino acids. Get that? And that's what makes your muscle different than that muscle. And so if you were to say, I will tell you something that's interesting about proteins. They have a primary structure. What is the sequence of amino acids? And then they fold in on each other. And we'll talk about that too. And then they have a final structure they fold into, and that's the protein. And so if you study proteins in college or as a PhD student, you would have to be really careful, because if you let the pH change very much, you'll denature your protein. It won't work anymore. If you change the temperature too much, you'll denature it. When I cook an egg, I want to denature it. All the egg, you know that gooey stuff? Not the yellow stuff. You know that all the gooey stuff? Why does it turn white? You're denaturing all those proteins, and now you can't even see through it anymore, can you? That part of the egg is almost pure protein. If I were to take some nitric acid and I open up an egg here and just throw it on there, it would turn white. I didn't cook it. You just denatured the protein and made it look different. And so if you're doing studies on this, you have to be careful. What do you think you might need if you're doing an enzyme study? It starts with a B. You have to watch your temperature. So you always have ice and stuff like that. You don't want to get too warm. And there's another thing you want to have with you. Starts with a B. What do biologists who study enzymes always have? Somebody said it.
Base.
No. BU. F. Buffers. Why do you need buffers? I can't let that pH change very much. I'll denature my protein. See that? There's some amazing stuff going on in the scientific world if you want to do that. The COVID vaccinations, those are all messenger RNA based. How in the heck did we figure out enough to figure out how to change the sequence of messenger RNA to make a vaccine against this? And right now, I think we're at the point where we can make a vaccine. It used to take 10 years. Some of these vaccines we can make in under a year because of that. So maybe you said, well, I'll get a little tired of this organic chemistry. Well, thank God not everybody did. Not everybody got tired. Oh, medical school, that's a lot of money. You go to school for a long time. I don't want to do that. That's okay. But thank God not everybody chose to do that. You wouldn't have doctors. You wouldn't have scientists. Somebody's going to be a scientist. It just all depends on what your motivation is. What do you want to do? You know? Do you want to find that? You say, I really would like to find a cure for that. You're going to have to get into this chemistry and physics and biology. You have to get into it. All right, so here we go. What is the primary structure of a protein? What's the primary structure? And that's called the sequence of the amino acids. And I'm going to show you the amino acids. And I actually have you, I'm going to have you learn these. Hey, see those 20 amino acids? All right, see the little line I drew here? Okay, down here, they're polar. Up here, they're nonpolar. Pick your 3 favorite polar and your 3 favorite nonpolar, and I want you to learn those, okay? Maybe by Wednesday. Now, I'm not asking you to learn 20. And here's what I'm going to show you, that all amino acids have the same central, I'm going to draw it for you. This is your central carbon. I got you. And it's called an amino acid. So here, you're going to have N, H, H. There's your amino N. Here's your central carbon. Attached to that is an amino N. And then there's always a hydrogen here. They all have that in common. And then over here, you have COO. C, double bond O, OH. Oh, I get it. I get it. That's the acid in. Amino acid. Now, if you look here, they all have that in common. They pretty much all have this in common. So why are they different? What makes them different? And you guys haven't read this yet, right? They call it an R group. An R group is whatever's attached right there. Now look up here. What is the simplest nonpolar R group that you can have? Yeah, I'm going to learn that one. Glycine. Glycine is the only thing you put up here is H. Okay, so this right here is the only reason why the 20 amino acids are different. So everybody's going to draw that. Or I might draw that part of you, see? What if I draw that part for you? And you have to learn this. You said H, glycine. I got that one done. What's the next one? A methyl group? Oh, yeah. Oh, yeah. I'm learning that one. What's that one called? Alanine. All right. Ooh, that one was complicated. Well, that's kind of cute. Ooh, look at that one. That has sulfur in it. Ooh, yeah. Anyway, we'll pick another one. I will tell you that I'll do this for extra credit if you want to learn that one. But that's not one of the 3 you have to learn. If you want to do it for extra credit, I'll give you a chance to do that. Now let's look at the polar groups. Oh, okay. Here's one. Serine is CH2OH. And that's what makes it polar, isn't it? See that OH group at the end? That makes it polar. I don't know, that's a little complicated. Well, that's kind of cute. Maybe you want to do tyrosine. That has a benzene ring in it, doesn't it? There's extra credit. I'll give you extra credit for that one. Let's see, what's another one that's not, that's pretty easy. That's pretty easy. That's CH2SH, cysteine. Anyway, I want you to pick 3 polar and 3 non-polar you think you can memorize, and it's just pure memorization. I can tell you that right now. But now what I'm going to do is I'm going to show you well, how do they come together? How would one amino acid, how can you get an amino acid joined to another amino acid? If you think about it, here, I'll do a simple one here. How many more bonds would you like? None. Get away from me. I don't want to be on it. Why would I want to join with another amino acid? And it's not a matter of want anyway, it doesn't have a brain. But God made it so that amino acids can be joined to other amino acids. Now, what do you think is necessary to join 2 amino acids? Starts with an E. And if you're a biologist, what chemical has to be there to make one amino acid join to another? Starts with an E. N. Enzyme. But enzymes are proteins. A protein is used to connect other proteins together? That's right. That's right. Now look in your book. Look right here. In your book, do you see the amino acid part here? Do you see the amino and the acid? You ever see that? And here's another one. I put 2 amino acids together, and an enzyme will come up, and because of the shape of the enzyme, it'll attract this OH and rip the OH off of the carbon. And it'll rip the H off the nitrogen. And what does that form? Water. And so what kind of polymerization is this? Is this condensation or is it addition polymerization? Condensation. So if you remove water, meaning an H off of you and an OH off of you, then doesn't this carbon want to form another bond? You just took away my OH, didn't you? And doesn't this nitrogen want to form another bond? Oh, there we go. There we go. Oh, and they have a special name for that. What's the special name for the bond that holds amino acids together? And I know you haven't read this yet. It's called a what? A peptide bond. Now, if you become a biology person, what they'll say is that amino acids are joined together with a peptide bonds, right? And then you form a polypeptide chain. It makes sense to me now. It's a polypeptide chain. Now, you're not really a protein yet because all I've done is put the amino acids in the right sequence. Now I have to let them fold in on each other and make a shape. And then I'll have the protein. So I didn't understand that when I was in college. I didn't know any about this model building. I didn't know any of that. And so I just said, okay, I guess you want to call it a peptide bond? I better remember that. And a polypeptide, I never made the link. A polypeptide is a bunch of things that held together with peptide bonds. All right, so anyway, when you get done, you would have not 2 monomers, but you'd have a dipeptide, wouldn't you? Dipeptide. That's what that dark print word is. It says it's called a peptide linkage, and blah, 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 blah. It says here, the order or the sequence of amino acids in a protein chain is called the primary structure. Now look at this right here. Here's lysine, alanine, leucine. And okay, that's the sequence of those 3. But again, how many ways can I put 20 amino acids together if you can use one as many times as you want? Could I have leucine, alanine, leucine, lysine, lysine, leucine? Wow, I could have millions of combinations. Isn't that right? Anyway, so that's the primary structure. Now let's take a look at this. It says here, write down the sequences of all the possible tripeptides that will be composed of the amino acids tyrosine, histidine, and cysteine. Okay, let's try. How about A, B, C? Could I have A, then B, then C? What's another sequence I could have? What other sequence could I have that starts with A? I could have A, B, C. What else? A, C, B. Okay, now start with B. B, A, C? B, C, A? Now start with C. C, A, B? C, B, A. I got it. Okay, how many are there? How many? 6? Okay. Do you remember this in math? Have you ever taught this little thing? Exclamation point thing? What's that called? Oh. What does 3 factorial mean? 3 times 2 times 1. How many is that? 6. How many amino acids you have? Here, do that. What is it?
2.433
To the 18th?
times 10 to the 18th.
Okay, that's how many with just 3. With just 3. Yes? Anyway, and some, a lot of amino acids, you don't even call yourself a protein until you get to be 70, maybe 70 amino acids long. So you can see that God made almost an unlimited number of proteins that can be made. All right. Oh, is that what you got?
Yeah.
Okay, that's what I had in the book. I forgot that was in the book also. Well, anyway, so then what they do is, imagine you have these amino acids in a long chain. And did you notice that, let me show you something here. Did you notice that this R group is different than R group? And see how the R groups are different? And some of them like have sulfur in them, and some of them are polar, and some are non-polar. What's going to happen if I have a long chain of amino acids and that these little side R groups are sticking out? What's going to happen when... Is it possible that I could be attracted to you, and maybe down the road you're attracted? Could I actually bend myself into different shapes because of that? And again, I'm not sure how we figured this all out, but we figured out a lot of stuff. And here's one. One thing that happens a lot with amino acid chains or proteins is that they'll form a helix. You know what a helix is? Spiral. Okay? And they actually call it alpha helix, meaning if I look down the row, it goes this way. It's a right-handed helix. So one of the shapes that we have in the secondary nature of a protein would be things like helix or a pleated sheet. So let me show you what that looks like. Now, I know you haven't read this, but if you go home and read this, I think it'll make more sense to you. This right here, this portion here is showing like a helix going around here, and this is like a folded piece of paper, like an accordion. And I didn't do this, but they call that a beta pleated sheet, or a pleated sheet. You know what pleats are? Pleats are like folds. And so what I would ask you to do is tell me 2 examples of secondary structures in proteins, and you'd say helix and a pleated sheet. You okay on that? Now, that's what they're trying to show you here. And then there is a tertiary. There's a tertiary structure, and the tertiary structure means that we then will fold in on each other. Now, I think, there it is. I need you to go, and we're going to find another page here, and it's page 974 and 976. 974, 976. And I think this is pretty neat. But 974, 976, there you go. 974, 976, 976. Okay, what's this? The heme group. The heme group. Where have you heard of that? Heme. H-E-M-E. What's a heme group? What is it? Hemoglobin. Okay? So here's this structure called hemoglobin. And notice that this is found in everybody. You have thousands of these in every red blood cell. Got that? Hemoglobin. And, you know what's in the center of every hemoglobin? This is pretty neat. What weird element is found in the center of hemoglobin? Iron. You know how we talk about iron and blood? Okay, that's why. And I don't have chlorophyll, but do you know that chlorophyll is very similar to this? And yet, what's in the center of every chlorophyll molecule? magnesium. Now, I think, and I'll show you one more and then we're done. This right here is trying to show you, all right, I better let you go. All right, thank you. Hey, if you do, you can just kind of listen to me for all day tomorrow, or if you want to read a little bit ahead of time, I think it'll make more sense to you.